Imagine a world where life is not born but meticulously designed in a sterile laboratory.
Scientists are now moving beyond reading the genetic code to actively writing it from scratch. This revolutionary field is known as synthetic biology, and it is changing everything we know.
At the heart of this movement are high-tech labs filled with automated DNA synthesizers.
Researchers are no longer just editing existing organisms like we do with basic genetic engineering.
Instead, they are constructing entire genomes using chemical building blocks in computer software.
The journey began in 2010 when the Craig Venter Institute created the first self-replicating synthetic cell.
They named this landmark creation Synthia, and it proved that human-made DNA could boot up a living organism.
Since then, the race to build more complex forms of life has accelerated across the globe.
These labs operate like biological software factories where DNA acts as the ultimate programming language.
Scientists use four chemical bases represented by the letters A, C, G, and T to write biological instructions.
Once the digital code is perfected, a machine assembles physical strands of DNA to match the design.
This process allows researchers to create organisms with functions that never existed in nature. One major goal is the creation of a minimal genome which contains only the absolute essentials for life. By stripping away unnecessary genes, scientists can create a blank canvas for custom biological features.
Imagine bacteria engineered to eat plastic waste or clean up devastating oil spills in our oceans.
Other labs are focused on creating synthetic yeast that can produce rare medicines or sustainable biofuels.
This technology could replace massive chemical factories with small vats of programmed microbes.
Some researchers are even exploring xenobiology, which involves life forms using non-natural building blocks. These alien organisms might use different amino acids or even a 6-letter genetic alphabet.
Such breakthroughs suggest that the definition of life is much broader than we previously imagined. Inside these facilities, the atmosphere is a mix of intense focus and futuristic ambition.
Rows of liquid-handling robots move with precision to mix delicate chemical cocktails.
Huge computer clusters run simulations to predict how a new synthetic organism will behave.
Every single experiment is a step toward understanding the fundamental mystery of how life began. However, the ability to create life from scratch brings up profound ethical questions for humanity.
Critics worry about the potential for accidental releases of synthetic organisms into the wild.
There are also concerns regarding the security of biological data and the risk of misuse. To address these fears, many labs implement strict containment protocols and digital kill switches. These safeguards ensure that synthetic life cannot survive outside of its controlled laboratory environment.
Despite the risks, the potential benefits for human health and the environment are truly staggering. We are looking at a future where we can grow organs for transplant or cure genetic diseases at the source.
Synthetic biology could also help us create crops that thrive in extreme climates or resist all pests. This would provide food security for a global population reaching 9 billion people.
The cost of synthesizing DNA has plummeted over the last 2 decades, making this tech more accessible.
Small startups are now competing with giant universities to unlock the secrets of the cell. We are witnessing the dawn of a biological revolution that mirrors the computer age of the 70s. Just as we learned to program silicon chips, we are now learning to program living matter.
The boundary between the natural world and the engineered world is becoming increasingly blurred.
Some visionaries even dream of using synthetic life to terraform other planets like Mars. Engineered microbes could produce oxygen and soil to prepare distant worlds for human arrival. Back on Earth, researchers are trying to resurrect extinct species using fragments of ancient DNA.
While we are not yet at the level of Jurassic Park, the science is moving remarkably fast.
Every day, new papers are published detailing the assembly of larger and more complex genomes. The ultimate dream is to build a multicellular organism entirely from laboratory-designed components. This would be a monumental achievement that redefines our place in the universe. We must balance our curiosity with a deep sense of responsibility for what we create. The future of life on this planet is no longer just a product of random evolution. It is now a collaborative project between nature and human ingenuity. As we step inside these labs, we are looking at the very first blueprints of the future.